going to speak about, as requested, is familial Parkinson's disease. Now, normally I could do this right off the top of my head because up until 96, we were always somewhat doubtful as to the role of genetics. In other words, our endowment from our parents, what's born in the DNA, and that makes us similar more or less to our parents. So I will summarize what I knew up until 97, and from 97 to the present there have been a lot of genetic advances, and we've identified at least 8 genes that if they don't immediately increase the risk will definitely cause the disease. It's oftentimes these genetic mutations, these changes in the genes that I'm going to be describing, don't necessarily mean you will develop disease, but it will increase the likelihood significantly that you will develop the disease. In other words, there are a certain number of risk factors that we've known about for the 20th century. So what the biggest risk factor that we've always thought, and we've always thought that Parkinson's disease is caused by environmental factors. And why was that? Why did we think that? Well, there was never really very good family histories. We always knew that maybe an individual with Parkinson's disease may have an uncle or an aunt with it, but it didn't seem to cover every generation. So we didn't think of classical inherited Parkinson's disease in the classical manner that I'll describe when I talk about autosomal dominant inheritance versus recessive inheritance. Just a few simple ideas about genetics. Otherwise, why talk about familial Parkinson's disease if you don't understand genetics? And of course, you will not understand genetics after I'm done, but you have a better understanding. So here's when I started in Parkinson's disease as my major focus of interest was because there was a report that came out in 1983. I was still in my neurology training and had decided to become a movement disorder fellow to focus on Parkinson's disease. I had no reason to. But there was an epidemic, a small epidemic in San Francisco, where young people developed Parkinson's disease pretty quickly. These people, it turns out, were IV drug abusers, and they were buying a material called China White and using it like heroin, intravenous injections. And a number of cases were coming into the emergency room at the various hospitals in the Bay Area, and it was really striking to see 20-year-olds, shuffling gait, drooling, stiff limbs, exactly the picture of Parkinson's disease. And we've seen this before in the 1950s. Many of you hear me talk ad nauseum. I talk about the breakthrough about dopamine hypothesis was from studying psychiatric patients who were treated with major tranquilizers, the new ones that came out in 1950, phenothiazine, lorazine. And those people would develop, young people would develop Parkinsonism, but it was reversible. You took away the drug, Parkinsonism would disappear. We still have drugs that cause Parkinsonism that's reversible. For example, Reglan is something to help empty the gut, can cause Parkinsonism. If you stop using it, it'll go away. Some of the anti-nausea drugs can cause Parkinsonism. So what I do when I review perfenazine that's added with antidepressants can cause Parkinsonism. So we still have drug-induced Parkinsonism. And so having trained as a pharmacologist, in other words, I actually completed my PhD in 1976, well before I went to medical school, and I worked on opiate dependence. So I was really intrigued when these young IV drug abusers developed a permanent Parkinson's disease, and they're using an opiate drug, an underground synthetic drug. So I said, wow, this is something worth studying because it'll give us clues as to environmental chemicals, toxicants that can cause Parkinson's disease. And so the idea then fit with our current ideas about the cause of Parkinson's disease, current up until in the 20th century, that it's caused by environmental factors. So that's how I got into the study of Parkinson's disease, and I spent 15 years in researching environmental factors that can destroy dopamine neurons and mimic or cause Parkinsonism. So there are many that have been found. One of them is the heavy metal manganese, certain pesticides, etc. But even before then, in the 1915 to 1926, people develop a post-encephalitic Parkinsonism. And so again, we found that it wasn't heredity that was causing the illness, it was a virus. For the longest time, people thought all Parkinson's disease was a result of certain kinds of virus infections. And then we found that chemicals can cause Parkinson's disease. So the hereditary aspects fell by the wayside until 1997. And this kind of research into the genetics was always kind of percolating amongst the researchers because we knew that individuals who had a family history had a somewhat higher risk. For example, the lifetime risk of Parkinson's disease, it's a simple way to remember it, by the age of 70 is 1%. So 1 of 100 people who are 70 in that decade of the 70s will have Parkinson's disease. So that's what you can expect, 1 in 100 chance. But if you had a mother or an uncle or a first-degree family relative with Parkinson's disease, your risk was doubled. That seems big, right? You have a 2% risk. But still, even with that familial risk, you had a 98% chance of not developing Parkinson's disease. So again, people weren't that excited about exploring the genetic aspects of Parkinson's disease until the techniques of molecular genetics got better and better. Refinement in these techniques allowed us to look at all kinds of variations in genes called polymorphisms in specific genes. And anyway, in 1997, there was a report by his Greek name. It's a funny name because it's like polymer, right? Polymorpoulos. So Polymorpoulos was the first author. And they described, with a big team of researchers, an autosomal dominant familial pedigree of Parkinson's disease. And autosomal dominant meant that every generation would have the illness. That each child of one of the affected individuals had a 50% chance. Now, here's your basic understanding of genetics. You know that you need a mother and a father as a human being. You get half your chromosomes from father, half the chromosomes from mother. Chromosomes are the organelle where all the DNA sequences are kept and preserved, like a safe deposit box. And in the process of making sperm, and in the process of making egg, the chromosomes divide in half so that the newly formed fertilized egg has a right number of chromosomes, which is 23 pairs of chromosomes. So that's the fundamental concept. So every cell has 23 pairs. Notice I say pairs. Just like we have a right and left nostril, we have a pair of every gene. And in an autosomal dominant disorder, you just need one of that pair to have a mutation, to have a problem in the code for that illness to be expressed. Even though you might inherit the normal gene at the other half, right? If you have that mutated gene, you're going to have it. That's called dominant. And autosomal means it's not related to the sex chromosome. Remember, there are 23 pairs of chromosomes. The 23rd pair is X and Y or X and X. And that determines one's sex. So a male is always an XY, and a female would be double X. Mutations on those sex chromosomes will result in sex-linked inherited disorders. And Parkinson's disease, all of the Parkinson's disease mutations I'm going to quickly survey are autosomal. They're on one of the 22 pairs of chromosomes, but not on the sex chromosome. In other words, even though there's a slightly higher number of women with Parkinson's disease, it's not genetically determined. It may be that women have some protective hormones that may result in the final number, but it isn't that women can't inherit the illness. They do. So that was how it was in 97. So it was very exciting to find this mutation. It was called the alpha-synuclein mutation. You've probably heard of alpha-synuclein. I never heard of it till 97. It was an obscure protein that's involved in normal synaptic activity in brains. Synapses are where neurons communicate with each other. And it was very much involved in vesicular or release of neurotransmitters. But when it was mutated, that protein tended to form an abnormal three-dimensional conformation. It would accumulate in the cytoplasm and would grow and then was a component of Lewy bodies. Now, Lewy bodies we knew since 1913 is the pathological hallmark of Parkinson's disease. People with Parkinson's disease, when their brains were examined after death, were found to have these abnormal proteinaceous materials that would accumulate because they couldn't be further digested by the normal proteasomal system that breaks down proteins for recycling. So the alpha-synuclein protein aggregated and accumulated and resulted in the demise of neurons. Wherever the neurons were that contained this alpha-synuclein, they would eventually dysfunction. It turns out that because of that discovery, they were able to start looking at alpha-synuclein, which is a more sensitive marker than the old Lewy body stain. The Lewy body stain is an early 20th century stain. It was a little pink color of a spherical inclusion that you would see under a microscope. But if you looked at alpha-synuclein with a much more sensitive way, you'd see a lot more Lewy bodies. And so there was then a study of the distribution of Lewy bodies from the various early stages of the illness till death in a famous study by Brock and Brock, where they looked at 150 brains in their brain bank and compared the clinical data from the brain bank, and were able to show that the first Lewy bodies appeared in the olfactory bulb, structure where the the smell nerves end, or in the brain stem down in the vagus nerve. And the vagus innervates the gut. So right away, finding Lewy bodies there early on made you think, it's probably Lewy bodies are coming in from the outside. Something that triggers Lewy bodies is either breathed in, travels in through the olfactory nerve, and which is a fascinating thing because that's what I'm working on now. Using that highway into brain, you have direct access to brain if you get it right into the olfactory nerve. Many viruses cause disease that way. They enter through the nose. They enter through the olfactory nerve, not through the vascular system of the nose. That's old hat. We've known that people have snuffed nicotine snuffs, they've snuffed cocaine, but that's through the vascular system, where the drug will quickly get into the capillaries of the nose and quickly circulate and get to the brain. I'm talking about nerve to brain. So the Lewy bodies gave you a hint that that was going on, but even more interesting, everyone began to think, well, maybe all Parkinson's disease, because the people who had this hereditary form, autosomal dominant Parkinson's disease, known as Parkin1, or the alpha-synuclein mutation, they have the same pathology as, quote, idiopathic Parkinson's disease. And idiopathic Parkinson's disease is the clinical syndrome of slowness, rigidity, cogwheel rigidity, that has no explicable cause. That's why it's called idiopathic. You remember, I tell you, as I tell my students, the doctor is an idiot, the patient is pathetic. Idiopathic. That means we don't know. We don't know what causes it. There are many idiopathic diseases. Once you find a gene that causes it, then it becomes familial Parkinson's disease. So we wanted to see, and there's a wonderful review of familial Parkinson's disease genetics, clinical phenotype, what it looks like clinically when the patients are examined, and the neuropathology in relationship to, quote, idiopathic Parkinson's disease. So if you looked at alpha-synuclein mutation, by the way, this is an extremely rare mutation. It was found in an Italian-Greek family. Large family, but that gene that was originally found has not been found anywhere else in the world, and no one with idiopathic Parkinson's disease that has been tested has that mutation, unlike some of the other mutations that I will speak about. However, more recently, there are a number of mutations of that gene that were a little different. In Germany, for example, they found individuals that had duplicated that gene. They had an extra copy of that gene. Or some of them had triplicated the gene. Now, when you duplicate the gene, the clinical phenotype, how it presents, is earlier than idiopathic Parkinson's disease, which is usually presents at age 60. It's when it's diagnosed, on the average in the United States. But these people with a duplicate form of alpha-synuclein had an earlier onset. But if you looked at their brains, they have Lewy bodies. And those Lewy bodies contained alpha-synuclein protein. So that was a big breakthrough. The next big genetic marker was another form of hereditary. The next form of familial Parkinson's was called the Parkin mutation, P-A-R-K-I-N. And the Parkin mutation had a pattern of inheritance that was autosomal recessive. That means that you need to have a copy of each of the genes for the Parkin gene, one from the father, one from the mother, and you need to have both of the mutated alleles or genes present to develop illness. So in those cases, you need a mother and a father. Typically, they're first cousins. Then they're very likely to each carry that mutation. And the first discovery of this was in Asian populations, Japanese especially. They were first reporting this, and there's a lot more perhaps inbreeding to some degree. So you'll see this autosomal recessive. Clinically, it was very different. It looks the same. They have rigidity, slowness, but it tended to be young onset before the age of 40. So Parkin mutation results in young onset, but otherwise has the same progression. They develop dyskinesias with levodopa, they get better with levodopa. So the big difference between Parkinson's disease, which is younger onset, is in the postmortem brain study. They don't have Lewy bodies. And that caused a turmoil. How could it be that this is Parkinson's disease, but we've defined over the 100 years that you have to have Lewy bodies in the brain to call it Parkinson's disease? So now we're redefining the nature of Parkinson's disease based on postmortem analysis because of that. And now there are about 9 different mutations. I'll go over a few of them. And they're named Part 1, Part 2, Part 3, like a parking lot. So now we have all these genes that we've identified based on familial studies. We have a cluster of people with Parkinson's disease. You get their blood samples, you analyze it, and you find new genes. So the next gene that was found was called, autosomal recessive Parkinson's disease. The Parkin is pretty common. I don't see it that much, but in general, some of these mutations, when they occur, result in a younger onset disease. So what I always suspect, if I have someone who's 26, 28, 30, when they first develop Parkinson's disease, I make sure they're not taking drugs that cause Parkinsonism. And they tend to have a family history. They tend to probably have more of a genetic component. Let me pick up the next one. Here's, like I said, this is it. This is what I knew up until now. Now I just read a review, and it's expanded what we know about the genetics of this illness. And many of these genetic tests are available through commercial labs like Athena Lab. So if I ever have a patient who's younger onset and there's a family history, I might order a genetic test to see which of these 8 mutations it might be. The next one is called a mutation in the UCHL1. And this is also autosomal dominant, initially found in a German family. And it, let's see what it described. And it's interesting because that UCH1 is a protein that's encoded by the gene that is also a component of Lewy bodies. So you can see it in Lewy bodies. And it co-localizes with Lewy bodies that localize in dopaminergic neurons and other cells. It's interesting because that protein is one of the most abundant proteins, the normal protein, in the brain. About 1 to 2% of all proteins in the brain are this one. But when it's mutated, it can cause the demise of dopaminergic neurons. They form abnormal. Again, that's not a very common one. The other one, this is kind of easy to remember, age of onset. It's called Pink1. You know that musician Pink? Very popular amongst young people, was anyway. Well, people who have Pink1 mutation become rock and roll dancers. No, they have an early onset Parkinson's disease. Okay, Pink is, I don't know how many of you know Pink, but Pink is not Frank Sinatra. Okay, so, and it's found in various ethnicities around the world, the Pink mutation, also known as Park6. The thing about it is they have an early onset, and they have really early onset of levodopa-induced dyskinesias. So you'll see this very early on. So a lot of people like Michael J. Fox, Muhammad Ali, they had young onset. They were 36, 28 when they had the illness, and they developed levodopa-induced dyskinesia really early. I suspect they had the Pink1 mutation. The other gene that became with Park7 was called DJ1. I'm not going to get into every detail. The next one is Park8, LRRK2. Now, LRRK2 is a fascinating one. LRRK is like the bird, but it's spelled LRRK2. And what it really stands for, it's a mouthful, but I like to say it, leucine-rich repeat kinase. It's an enzyme. And a mutation in that enzyme results in pathology that causes Parkinsonism. What's fascinating, there's another name that they called it, dardarin, meaning tremulous. I guess, I don't know if it's a Hindu word, but someone named it dardarin. That protein, instead of saying GLARC2, is dardarin. And these mutations, fascinatingly enough, are the most common mutations in sporadic Parkinson's disease. In other words, people with no family history of Parkinson's disease, if you start looking for LARC2, you'll find a variation of LARC2. So we know that mutations in LARC2 may not actually cause the disease, but it will increase the likelihood of causing the illness. And that's one of the most frequent variations of the gene that increases the risk. And we have PARC9, which is ATP, 13, et cetera, et cetera. So I'm going to end that little overview by underscoring what I tried to point out. I still believe that that environmental exposures to specific toxicants can destroy dopaminergic neurons and produce a syndrome of Parkinson's disease. There's clearly a very important role of heredity in causing familial Parkinson's disease, and some of them, frankly, cause it directly, like the alpha-synuclein, the Parkin. Some of them just increase the risk of developing illness. And what I often think is this interface between environment and genetics has to do with how we metabolize environmental chemicals, like certain pesticides. And some of my early work, I found that a pesticide, dieldrin, which is used for termites, it was banned in 1971 because it could cause liver cancer. And it was banned, but when I did a study in 1993 where I collected brain tissue from Parkinson's disease brains and age-matched controls, I found really high levels of dieldrin, even though it hadn't been used since '71. It's very fat-soluble, this pesticide, and accumulates in the brain, especially in those deep gray structures responsible for automatic execution of movement. And yet, when I went to a town in Clarksville, Mississippi, I was still at the University of Miami, and I had a local physician who said, we have a huge cluster of Parkinson's disease, and they're all cotton farmers. You want to do a study? I said, please document their diagnosis. Send me blood samples of people who work in the cotton business, cotton picking, packing, etc., spraying the crop, versus those age-matched controls who never work with cotton. And guess what we found? That people from that town, everyone had really high levels of dieldrin, even people that didn't have Parkinson's disease. And so what I wondered is it that if you have certain genetic endowment that allows you to metabolize this pesticide, you won't develop the illness, but because everyone's exposed to high levels. By the way, this may change your attitude about buying Chile grapes from Chile. They still use this darn thing in other parts of the world. This is one of the weird hypocrisies. Again, I shouldn't talk about it, but we ban certain things here, but we can sell them abroad, and then we import some of the products. And so this stuff stays around in the biosphere a long time. So that's why I'm kind of an environmentalist. Protect our environment because it comes back and bites your butt eventually. So I'll take questions. And then I got to end early because I have some guests that I have to speak to. Go ahead.
Doctor, you mentioned some Parkinson-causing drugs, and you mentioned Reglan.
Yeah.
Now, I take a generic Reglan.
Same.
And I didn't have,
You may not, but here's what happens when you're this age of 40, which is now, it seems like a spring chicken to me, anyone who's 40. But from the age of 40, you start losing dopamine neurons. And so that you are more susceptible to the effects of a dopamine receptor blocker when you're older than when you're younger. So you might have been 50 when you used Reglan, but if you're 65 or 70 using Reglan, you could develop a transient Parkinsonism. So yeah, I wouldn't use it. Use Nexium instead.
I have gastroparesis.
Well,
and that's why I get it. That's why I take it.
Yeah. Do you have Parkinson's disease?
Yes.
It's not a good idea to take it. You have to find something else. Talk to your GI doc. Say that Reglan is not something that people with a dopamine deficiency should be taking because it probably worsens your condition, your clinical features. You gotta get off of it. Another question? Yes.
If somebody has Alzheimer's in the family, does that increase the possibility of their children getting Parkinson's?
No.
Because
Alzheimer's has a whole nother story. The pathology in Alzheimer's is very different. It has different proteins that accumulate called beta amyloid and neurofibrillary tangles. There's definitely hereditary, there are genes that can increase the risk of Alzheimer's disease, just like there are. But if you have Alzheimer's disease, it doesn't increase your risk of Parkinson's disease. However, let me finish why you'll see people with both Alzheimer's and Parkinson's disease, is that they're the two most common neurodegenerative diseases. So Alzheimer's is number one. There are more people affected by Alzheimer's than any other neurodegenerative disorder. Number two is Parkinson's disease. So it's not unusual for people with Parkinson's disease to develop aspects of Alzheimer's, and if you really test them carefully, you'll see they have Alzheimer's. Conversely, they have Alzheimer's. If you follow them carefully, a certain fraction of them will develop slowness of movement, rigidity, etc.
My question really was, are some of the genes that cause
No.
Alzheimer's, are the genes
Those are not the same genes. That's what I'm trying to say. They're completely different, completely genetic background. But you can see both in the same people because they're such common genes. You might have inherited genes from Alzheimer's ancestors, and you might inherit genes from Parkinson's disease. So if you're unfortunate to inherit both, you can eventually develop both clinical syndromes. They're very, very different. Okay, thank you.